Dear colleagues, nice to see you all here. Just finished a very good panel on market access, and if you missed it, the conclusion was there are no innovative payment models. It's going to be lump sum. So that means that you need to have a product that you can make relatively affordable fashion so that you have a proper margin when you start selling it with a lump sum model. Maybe a lump sum will with risk sharing, but anything more innovative seems to be not really in the cards. Sorry if I made that a little bit too easy, that conclusion after 45 minutes of a panel. We are a UK public company, and so that's a disclaimer. And we work all in cell-based therapeutics. From my initial point, we work in allogeneic technology, so that means that we have the opportunity to have what I would call a normal pharmaceutical margin, meaning 10% or less cost of goods from the prices that we think would be reasonable and correct in the marketplace. We work in significant indications. All of them have a large potential if we are successful. We have a clinical stage therapeutic portfolio, retinitis pigmentosa, motor disability resulting from stroke, and also for CLI. And we're working on research on exosomes, something that you heard of earlier today, and it's an emerging field in research. We're a very well-backed company. Woodford Investment Management is our biggest shareholder. We did a relatively large fundraise last summer, which means that we have ensured our future in that we can develop and continue all these programs that we have set out to do. We're sitting on $86 million or so in cash, so it gives us the opportunity to do what we set out to do. We have two different cell technologies or platforms. One is human neural stem cells, so that's the CTX cell line. That is our stroke program, as well as the exosomes. We harvest exosomes when we manufacture these CTX cells, so these are our human neural stem cells. Our second platform is our human retinal progenitor cells, so these are retinal cells for retinal indications. This was something we have in-licensed from Harvard Medical School, from Skeppen's Eye Research Institute some years back, and we're very excited about that program as well. We very much see CTX as a pharmaceutical product. It is obviously GMP validated. We have made it into a cryopreserved formulation. We have 6 months shelf life. That obviously could be extended as we go forward, but it makes life a lot easier in clinical trials if you have a 6 months shelf life. Our phase 1 study was done with 8 hours shelf life, so that makes it a little bit harder. That means you need to have your clinical trial center very close to your manufacturing site, which is what we did. So life is a lot easier with cryopreserved product. The pipeline, we have 2 different cell therapies and 3 different indications. And on top of that, we have exosomes, which is a non-cell product that's in research. And I'll go through these programs in more detail. I'll just throw up quickly the management and board of directors, and the point of that is that we have built a full management team because we have a very ambitious program over the next few years. Chief Medical Officer and Head of Regulatory Affairs we added about a year ago, and also our board. We just added some really great research firepower with Dr. Michael Owen, who came from GSK. So we have a really great team backing this. The retinal platform is based on human retinal progenitor cells. We hope to rescue existing photoreceptors in patients and maybe also build new ones. You're born with all the photoreceptors you'll ever have, and what we try to do is actually to rebuild photoreceptors in patients who need that. The first indication we're working on here is retinitis pigmentosa. So that is an inherited degenerative eye disease. These patients lose their vision gradually over time. It starts with losing their peripheral vision and their night vision, and over time, they will lose their vision completely and go completely blind. It is an orphan indication, but it's a very large orphan indication, 275,000 patients in the US and Europe combined. So this is the first target for this product. We have an orphan drug designation in Europe and the US, and we also have fast track designation from the FDA in the US. So we have started here a clinical trial. That clinical trial was based on these enabling preclinical data. We were able to show in animal models that we can improve visual function in this rat model, as well as we could show that we were able to extend the cells into the ganglion layer, which means full integration of the cells. That gives us the hope that we can actually restore these lost photoreceptors. But obviously more important is the clinical program. So we have started a phase 1/2 study at Massachusetts Eye and Ear Infirmary in Boston. Under the PI is Dr. Eric Pierce, who is one of the world leaders in the field, so we couldn't be in better hands than in his clinic. The first part is a traditional phase 1 study, 3 different doses, 3 patients in each dose. You basically wait a month between each patient due to DSMB meeting will happen after every single patient. We are now in the middle of that phase 1 portion, so that's ongoing. And then we will go into a phase 2 portion, which will be an additional 6 patients in what we expect to be the highest dose, which we expect to be 1 million cells. So these are implanted subretinally. You can see our graphic of how that's done. You need steady hands, so I shouldn't be doing that. The cells are implanted into the back of the retina. The retina is detached temporarily. The cells are implanted. This is day-to-day work if you're an eye surgeon. So this is kind of standard stuff. It is, of course, a one-time therapy. So it's a one-shot therapy. And we're very excited about this program. Our most advanced program is in stroke. And I shouldn't say stroke, because really it is motor disability resulting from a previous stroke episode. So we're not treating the stroke, we are treating the consequences of the stroke. Motor disability is one of the biggest issues following stroke. Stroke is the largest cause of adult disability in the West, in the Western world. There is no pharmaceutical treatment options except clot busters in the first few hours after treatment. Hopefully, Athersys will launch a product for the acute stroke. They are working on that one, so hopefully that will happen. That will still be within the first 48 hours. What we're talking about here is chronic stroke. So we're talking months or even years after stroke. We did a phase 1 study in 11 patients. These patients, this was 4 different dose groups, and we followed them for 24 months. That data was just published in The Lancet in August. And the conclusion was, first of all, no cell-related or immunological adverse events. And we do not use immunosuppressants in any of our clinical trials, so it's always important to keep an eye on that. And we have encouraging efficacy result across many different measures. There are so many different measures to actually measure clinical relevance or clinical impact in stroke. So we had a lot of measures. Poor patients had to go through a lot of testing. That has led us to go into a phase 2A study. That study has now completed recruitment, 21 patients. We used the highest dose from the phase 1 study, which is 20 million cells. The treatment window in the study protocol is 2 to 12 months. In practice, it's 6 to 12 months. The patient has to prove to be stable for 2 months after the stroke to get into the study. And even if the clinicians say that the patient is stable, it's not going to improve any further, very often the patients themselves think that they might improve. So they're not quite ready yet. So usually you need to give the patient a couple of extra months before they kind of given up hope that they will improve anymore. Which is good for us because we want patients who are very, very stable because we want to, whatever results we see, we would like them to come from the treatment and not by chance. We don't want to fool ourselves. So this is now the 3-month efficacy data. And the reason why I say 3 months is in phase 1, we could see that patients would reach the maximum efficacy after 3 months, and that would be completely stable throughout the 24 months that we studied in phase 1. So this is our expectation now in phase 2 as well. So 3 months we expect to be able to pick up the efficacy. And you will know when I know, which is in December of this year, how that looks in this 21-patient phase 2A study. And based on that, we will go ahead with the program, most hopefully in a phase 2 or phase 3 study following that. This study will be controlled. The control is obviously a little bit tricky because we're talking here about implantation of these neural stem cells into the brain. So this is neurosurgery. Again, standard procedure if you're a neurosurgeon, but not if you're me. And so this has been impossible to do a placebo control until now because you really have to show that there's a hope of efficacy before you can ethically do that. But I think after this 21-patient study has reported, we should be able to go ahead with a placebo control study. Placebo control means it's a placebo surgery. The patient will be sedated. The surgeon will make a hole in their head. But the dura will not be penetrated. So, but the patient will wake up thinking he or she had the surgery, and that is the placebo control. And you can also have the measurements done controlled as well, so, or blinded as well. So it sounds quite invasive for a placebo, but it doesn't add any risk since you don't penetrate the dura. And this has been done several times before, so that's okay. We talked to regulators about it who are happy about it, a little bit less happy in Japan. But in the US and Europe, they're fine with that. So we're looking forward to the data in December and the next step after that, which will be a very interesting placebo-controlled trial. We have a second indication in CLI, which I'm going to skip more or less since we have only 2 minutes left. What I will say quickly is that we have a phase 1 study ongoing. You might wonder why we're using brain cells in the leg. And it is because of the mechanism of action is regrowth of new blood vessels. We can see that very well in animal studies. So we hope that these neural cells will grow blood vessels in the leg of these patients. This phase 1 study is ongoing. We will be quite careful in this program. And the reason for that is that it's a very difficult indication. Patients have a lot of comorbidities, and there are multiple new treatment options coming or emerging, so we'll take it quite slowly in this indication. We will be looking for the right subgroups in order to show what we can do. A quick look at the Exosomes program. This is very, very interesting, quite early stuff. We are harvesting the exosomes from our CTX GMP production line. We're spending a lot of time actually on the purification, the CMC part of that. So exosomes are nanosized vesicles that are important in cell-to-cell signaling. What we're hoping that these cells or these exosomes will do is to try to impact cells around them. So if you have, in a cancer indication, you have exosomes signaling to the malignant cells to behave normally, so to turn the apoptosis, the normal cell cycle, back on, which would be a huge impact in oncology. That's one of the still standing pillars that need to be torn down in oncology to win that fight, is to get the cell cycle back again. So cancer is one treatment option, and the other area of potential for exosomes is to use them as a vehicle to load gene therapy so that they can get to the right place. So we're working on those two. For the oncology program, we have selected a candidate called ExoPro, and this candidate has shown preclinically that it can inhibit glioblastoma cell migration as well as reduce tumor volume in animal models. So we are taking this forward, and this will be our first intended indication for exosomes. End of next year is the target for that. And we are getting we have a very nice grant from Innovate UK to help us, particularly around CMC, around exosomes, that we can really purify them and get them ready for clinical therapy. So our projected milestones you see here. So there's a lot going on, so we'll try to summarize it all here. For CTX, stroke phase 2 data coming by the end of this year. And then first half next year, we will initiate the next study. For HRPC, we'll have the safety data. So the phase 1 safety data will be the first half next year. And the phase 2 efficacy data will be the second half next year. The phase 2 portion goes quite quickly because we don't have all the DSMB meetings after almost every patient. So that part will go very fast. There are a lot of RP patients waiting to be treated, so there is absolutely no shortage of patients. The shortage is more surgeons' availability and the DSMB waits. So those are the key highlights of our milestones. And in summary, we have a differentiated platform which is cost-effective. We are looking at significant disease targets. We're well-backed and well-funded. And we have a strong team that can deliver on these ambitious targets. And with that, I say thank you for your participation, and thank you.